you all hear me? TAs could respond. Hello students, can you hear me? Maybe somebody can type in the chat box. Okay, great. Welcome all of you to today's live session. I hope you all are doing well. Everyone doing fine and going through the lectures. I hope enjoying and learning as much as possible from the lectures. So a lot of you have joined, so that's very good. And again, we have the same standard operating procedure today as last time. If you all have questions, then please type them in the chat box and also in the YouTube. One of the TAs is looking into the YouTube channel. So I think we can also check those out. He or she will relate to me. Okay. So I think maybe, some of you are registered for the exam also. And I think there has been an announcement on the forum. So I request all of you, please check all the announcements regularly, because some of the doubts that people have raised about the exams, the methodology and all that is posted there because it's a computer-based exam. And I wish all of you the best of luck for that. I will quickly share one of my screens so everybody can just see that also. Yeah, I think that will be good. So it's about the exam, and I hope you all will look into this in detail. I'll try to share my screen real quickly. I haven't tried that in this one, but let us see what comes up. You all can prompt me, okay, about what is it that what you're seeing right now. So is this, I think this should be about correct. All right. So I'm not going into the full screen sharing mode, but this is about the exam, okay? So all of you can look into that. There is some details available. And of course, it's a computer-based exam. You will have various options available to you, and you have to come with your IDs and so on. Exam is on 24th. So there's plenty of time for all of you to prepare. And we all, myself and the TAs, we wish you the best. And you can keep posting questions on the forum. So we will try to answer them. So, don't worry about that. And also I would request you to sort of keep your eye on the forum, because if we have any announcements or clarifications, or we want to provide you additional information, then we will do that via the discussion forum. So please just keep all of you regularly looking at that. I think it will be helpful for everybody. Okay, so TAs, please chime in and say something. Welcome the students, because I want to make sure everybody's audio video is working. Hello, are the TAs able to hear?
Yes, ma'am.
Yeah.
Hi, this is Devolina.
Yeah, Debolina, please adjust your camera again, your angle of your laptop. Yes, very nice.
I know.
That's good enough, yes.
Yeah. So, hi, this is Devolina. I welcome you all to today's live session. As ma'am said, keep on posting your questions. We will try to answer as many as possible. And this is Swagat.
Hi. Hi, this is Swagatnath. I'm one of the TS for the cell biology course. You can, we are trying to answer most of the questions in the discussion forum. So we can post the questions there, and we'll try to answer mostly.
So, over to you, ma'am.
Yes, one moment. So I do have certain questions that I would like to resolve. There were many questions in the forum, and I also looked up some of the earlier questions also, and still there are some pending. So I'm hoping we will have one more live session. So if anything gets remaining, don't worry. Of course, the forum is there for us to reply in writing, and then of course we can answer them there also if anything is left out. Okay. So, Deblina, would you like to go first, or I can also, I have some of the questions resolved also. So we can start. And I would like the other TA Swagat to please come here momentarily, because I want to see the YouTube questions also.
There are no YouTube questions yet.
No, please come here because I have another setup over here, right? Yeah. Yeah, one moment. I think, where's my Zoom window? Okay, I'm sharing screen right now. So can you set it up in this one? I don't think it will conflict. Yes, I also have them. Thank you very much. You can connect it to Wi-Fi. Yes, fine. So I will start in the meantime. Okay, some of the questions that came in the forum, which I think it will help all the students. And okay, Shine or Shiny. I don't know if she is there, but someone named Shiny has asked to please explain the chromosome conformation contact map in a bit simpler way. So you all might have come across this in Dr. Koshrin's lecture. And of course, this is a very important and useful assay to check out the three-dimensional conformation of a chromosome. Okay, so this is one of the questions that came to me, and I'm sharing screen, so probably you guys can see it also. So just let's look at the figure. So there are two versions of this assay that we will discuss, and we have discussed in the class. Okay, and so one is the 3C, okay, the chromosome conformation capture method, and the other is the micro C. You might have understood that from the lecture. So, thank you. Yeah. Right. So in this, here the schematic, I hope all of you can see this. Let's say this is a nucleus and there's chromatin inside. So you can see the nucleosomes and the DNA is wound around that. Okay, and then these fibers, chromatin fibers, they would be folded up in some particular three-dimensional conformation inside the nucleus that we don't know, but we do want to determine that. So to do that, first, this the cells, they are cross-linked. Okay, so why you do cross-linking? Because as you do the further procedures, you want to basically fix this conformation, because that's what you want to understand, what was the original conformation. So when we do different steps of the experiment, there are many steps, so you don't want things to be moving around. So you fix it. What fixing means, or cross-linking here, means that there will be cross-links generated between the DNA and the protein molecules with each other, and basically whichever molecules are close to each other, that is, they are in proximity, they will get cross-linked. Okay, so then that conformation is sort of, you can say, fixed. It's not going to move around much, although there'll be some flexibility still, but it's not going to completely lose its conformation. So after that is done, then in this technique, the chromosome conformation capture, the chromatin is digested with restriction endonucleases. So I hope you all know what are restriction endonucleases. I can't see the chat box here, but these are very popular enzymes, as you know. They are DNA endonucleases, they actually recognize a specific sequence in the DNA. So in any DNA, there'll be a particular sequence, a short stretch, it could be 4 base pairs, it could be 6 or even up to 10 base pairs, although that is rare. So these enzymes, they will cleave the DNA, but they only cleave at that specific sequence. So I don't know if you all can see my cursor. Is that visible? Or maybe I go in slideshow mode. Let's try that. Okay. Right. Oh, this is good. So now you all can see the laser pointer, this red dot here. Right. So we went ahead and do restriction endonuclease digestion of the chromatin. So now this enzyme will cleave wherever its recognition sequence is, which will be, the frequency varies depending on the restriction endonuclease you're using. So there'll be frequent cutters whose recognition sequences are short. Like if there's a 4 base cutter, it recognizes a 4 base sequence. So usually people will like to use that in this assay, because you want to be sure, wherever these contacts are there, you hope that your restriction endonuclease is cutting there. What's going on here? I did something. Yes. All right. So ignore these red arrows. I have no idea where they came from. So just, so it will cut at different points. Okay. And then after that, in this assay, what you're doing is I think someone's trying to do something. I'm really not. Please stop with this. Okay. So you have cut it, and now you're going to ligate it. Okay. So ligation, DNA ligase is an enzyme that will join the 2 ends of DNA. And it will do that to those ends which are close to each other. So because these were fixed in this 3-dimensional organization, the ends which are close to each other now, which have been exposed or cut with the restriction endonuclease, they, so the ones which are close to each other, they were the ones which were parts of the chromatin that was close to each other. Okay. So basically you go ahead and you ligate it. And so now you have these DNA fragments which are ligated. Here, 2 different chromosomes are shown, one in orange, one in green. And this part of them was near each other. Okay. So normally, for example, the nucleosomes which are right next to each other, they will, of course, ligate with some frequency, because the DNA on these, it's always going to be close to each other. Each one will be next to its neighboring one on either side. But here, because these 2 chromosomes were coming together in the 3-dimensional space of the nucleus, these get ligated. And so now you have a fusion DNA molecule which has been formed. Okay, and then you can go ahead and analyze. So what you need to do is analyze those junctions. So whichever junctions have been formed, which are parts of different chromosomes or even 2 distant parts of the same chromosome, you can infer that they were close to each other in the 3-dimensional organization of the nucleus. Okay, so but how do you do that? So there are many methods. So originally when this technique was invented, of course, one could do it by PCR. So if you're thinking, is this part of this chromosome close to this other one, you could have PCR primers, one on this one and the other one here, and you could do a PCR reaction. I hope all of you know what is PCR. It's one of the prerequisites of the course, a basic technique for amplifying DNA using primers and Taq polymerase or PFU polymerase. So if you get amplification, and you can also quantify that, so that tells how many of those fusion fragments were generated. First of all, whether or not they were there. If they were there, it means that at least in some of the cells, those 2 regions were close to each other. Okay, so this is a basic principle. And here, of course, they are doing sequencing. So therefore, you have these paired end reads, and you're going to look at the chromosomal coordinates of the contacting nucleosomes. So here it's explained because we are looking at the loops. The idea was to understand about the folding of the chromatin, which can happen by formation of the chromosome loops. So if there's a piece of DNA which is unstructured, it's not really folded into a loop, it will be somewhat like this. And as I explained, the two neighboring nucleosomes, they will most frequently, the DNA in those will cross-link and ligate with each other. Okay. And this will be true for each of them. So this one will be near this one, and this one will be near that other one also. So when you plot that along the chromosome coordinate map, so each of these is a sequence. So we have the sequence of the entire chromosome. Okay, and so the chromosome coordinates can be plotted on either side. And then we are seeing how many, quantifying how many of those cross-linked molecules were formed. Okay, so when you plot that, you get this straight line, because here the frequency of ligating to the neighboring one in this case are equal for all of them, and they will be happening best with the one which is nearby. So that's why as we go along the coordinate, chromosome position, that is, you can see that each of the neighboring ones uniformly will be ligating with the other one. Now, if you have a loop, of course, this will be true to some extent, because here also the DNA here is likely to ligate to this one, and so will this one, and so on. Okay, but you're looking for some outlier signals. For example, when there is a loop, then two different parts of the same chromosome, they will come together. I don't know if I have something I can explain you better. Yeah. So, like there's a thread like this, and if you have a loop, so normally these two regions are far away from each other. Okay. But if you have a loop, then they are close to each other. Yeah, you can see that. So when you do the cross-linking and the digestion and then you ligate, then these two sequences, they can be ligated to each other because they are nearby in the three-dimensional space. Got it? And if it was not folded, then they would be quite far. The chance that this will ligate with this one is very less. So when that happens, that's shown here. So here's a loop and the sequences which are the base of the loop. Even though in the normal chromosome, they were quite far away, here they are close to each other. So they can be ligated. And when that happens, so this DNA sequence is going to ligate with this one, and this one is going to ligate with the other one. Okay. And therefore, you will get these spots on either side. Now, they are away from the straight line, but you get quite a bit of ligation there. Got that? So this is the basic principle of this technique. And I think the question was about the chromosome conformation contact map. I'd like to move on because there's some more questions also. So this is what is being shown here. This spot corresponds to this linkage. And this is the actual result of the experiment for the entire chromosome. You do see this linear signal, and then you see these spots, to the sides, and they have a sort of a symmetry. And those, of course, are the ones which are probably distant, but they are close to each other in the three-dimensional space. And the technique used here in this lecture was micro-C. So basically, the micrococcal nuclease was used instead of a restriction enzyme. So the key difference between them is the restriction enzyme will cleave intermittently wherever its recognition sequence is there. But the micrococcal nuclease cleaves more frequently. So when you use such an enzyme, then the resolution of the technique increases because it's cleaving many places, and what ligation products you get is not limited by the distribution of the recognition sequences. Okay. So I think we have addressed that question quite well, and I hope you all can appreciate that. I think this is just, so when this was done, in fact, the main point was that in budding yeast using the 3C technique, loops could not be seen. But when micro-C was used, then loops could be seen quite well because of the higher resolution, because they used the micrococcal nuclease instead of restriction endonucleases. Okay. So I don't know if the TAs want to say something. In the meantime, I can, of course, continue and address some of the other questions also. So I hope this is clear. You all can write in the chat box. I think I hope Shiny is able to understand this, and the rest of you also. Okay, I think I will stop sharing my screen now. Okay. We'll be looking at some of your questions also. So let me just quickly go through. Yeah, there are lots of people bringing up points. So there are some pending questions also, and I need to sort of address them. So I'm just curious how many of you are there, about 40 or so. Great. So one of the questions is by Hridya Ramesh. Okay. Hridya, I don't know if she's there. So the question is, how does the position of chromosome regulate its expression? Because in one of the lectures where we were discussing about nuclear organization, this point was brought up. Okay. And it's an important point. Hridya is there. So there are many ways actually, but one of the very interesting ways that I've mentioned in the course, I think, and also we've done a little bit of work on that in my own lab also, is that depending on the position of that chromatin region within the nucleus, like where is it? Is it towards the inner side, internal part of the nucleus, or whether it's towards the periphery? The gene expression appears to vary. So many years ago, it was shown that the parts of the chromatin which are tethered to the nuclear envelope, which is the membrane surrounding the nucleus, and it has got many proteins also bound to it. So if the regions which are tethered or sort of linked or near the nuclear envelope, I would say attached to, bound to the nuclear envelope, they tend to be more repressed in general. And the regions of chromatin which are away from the nuclear envelope, that is away from the outer boundary or periphery of the nucleus and towards the center of the nucleus, they tend to be more expressed. And that's generally, so you can say euchromatin more likely to reside in the center of the nucleus. And the peripheral region is more enriched in repressed or heterochromatin-like regions. And most importantly, the ends of the chromosomes, the telomeres, they are actually attached to the nuclear envelope. They are tethered to the nuclear envelope. And people have actually shown that if you untether them, if you detach them from the nuclear envelope, so now they can float around and they can even reach the center, the same sequence, then the genes may get de-repressed. And if you're interested, you can look up one of the papers by my student Dipesh Kothiwal about that, where we have looked at this particular problem using a very interesting assay. So that's clear, I hope. The confusion about leading chromosome and lagging chromosome, this is completely different from leading strand and lagging strand, okay? Leading strand and lagging strand in DNA replication, right? That is leading strand where there's continuous synthesis and lagging strand where there is Okazaki fragments are formed and so on. That's a different term. But here, leading chromosome and lagging chromosome means when the chromosome is being segregated. The chromosomes, they are attached to microtubules, and the microtubules, they are pulling them away towards the spindle poles. Okay, I hope you all are understanding this part. So when the microtubules are pulling them, again, I'll make use of this. So assume that this is a chromosome, okay, and the microtubule is attached here. So anaphase is occurring, the chromosome separated, and now it's pulling it. Okay, so when it's pulling, so this would be the leading end, because this is the one towards the front. You can think of a boat, going in the ocean. The front part, which is moving forward, that is the leading end. And the lagging end is this. So this is lagging behind. This is more towards the center of the spindle. This is more towards where the metaphase plate was, where all the equatorial plane, where all the chromosomes are aligned. So that's what we mean here during chromosome segregation by leading and lagging parts of the chromosome. Got that? So basically, if condensation doesn't happen, then the chromosome shortening will not happen, right? So even when it reaches the end, then it will be this part. Let's say we are looking at, this is the end of it, and let's say the spindle pole is here. It has segregated, this is unfolded, not condensed. So if this is the equatorial plane, this part is still lagging behind, and it can be cut during cell division. And there are some mutants defective in condensin, in fissionase, where it actually happens, and they're referred to as cut mutants. Okay, but if there was condensation, then this will be folded up, right? And so it's segregated. It will be far away from the plane of division, which is here. So there's no risk of it being cut during cytokindation. So that's one reason why condensation is important. So I hope that's clear to you. Now, next question. I don't know if the TAs want to chime in and say something. All right. I think Priyamvada had a question earlier, which is why is DSB repressed in telomere and centromere proximal regions? So I think this may be pertaining to meiosis lectures, but this is a general point anyway. So I think this is probably because these regions are, there's heterochromatin, right, near the telomeres and in the regions flanking the centromeres. So they are more compact, they are tightly folded up. So any endonuclease you are using, I mean, in case of meiosis, there's 411, it may not be able to cleave efficiently here to generate the double strand breaks. And because of this, people have observed that recombination is repressed in these silent heterochromatin regions, such as telomeres and the pericentric regions. Okay, so Harsha, I don't know if Harsha is present, but was asking about how we can differentiate polyploidy from aneuploidy. So if you know the definition of these two terms, you can think along those lines. Okay, I don't know if Harsha is there, but right, so polyploidy basically is when the entire chromosome complement of the cell is there in multiple copies. So like in a diploid, you have two copies. A polyploid, maybe, if it's a tetraploid, it will have total like 4 times the haploid. That is 2 times what a diploid has. And it could be even more than that. So in that case, the entire set is duplicated, and depending on the number of times the polyploidy is there. Whereas in aneuploidy, you have maybe the loss or gain of a single chromosome, or you may have translocations or rearrangements. So a very small part of the genetic material is missegregated or missing or extra. So that is the key difference between the two, and therefore there are many techniques you can use. I mean, because polyploidy has got, in case of polyploidy, there's a lot more DNA in the cell. So even by techniques such as FACS, you could distinguish between those cells. Because in aneuploidy, it will be very close to the normal genomic DNA content. Whereas in polyploidy, it will be twofold or threefold or fourfold more, which you can easily measure using facts. And of course, you can do fish. You can even do a normal metaphase spread and just be able to see the chromosomes, and you will be able to tell the difference. Okay, so you can think about it. There are many more techniques. I just mentioned a few of them. Okay. Someone asked, what is ectheson? So this is, I think I was talking about this when talking about development. And this is a hormone which is, it stimulates metamorphosis and it regulates molting in insects mainly. So it's a regulator of molting and metamorphosis and reproduction in insects. I guess if any questions are left, we can address them next time also. So let me just see what else is there. Someone had a question. I think it's a bit confusing. I do want to discuss one assignment question. So, yeah, Rai Kir. I don't know the full name because it's not there in the email ID. Someone had a doubt about the 5th question in the week 7 quiz. And they feel that the answer should be both A and D, but there's no option. The question is the condensing complex tethers DNA. The question is you have to pick out the correct statement regarding the role of the condensing complex in chromosome structural maintenance. And, some various options, 6 options are given. And the correct answer was without condensation, the lagging chromosomes could be cleaved during cell division, of course. And the student feels that A might also be correct. The complex tethers DNA regions between sister chromatids to generate loops that compact the DNA. So I can understand the confusion because, in fact, condensin is also required for cohesion. I think I've said this in one of my lectures. And indeed, this is one of the known functions of condensin also. But when there's tethering of the sister chromatids to each other by condensin or by cohesin, loops will not be formed. So this part, the later part of it, is not really correct, and which is why A is not a correct option, although it's a little confusing. It kind of looks like it might be correct, but it's not. Okay, so I hope it clears that doubt. I don't know how many of you had that doubt, but definitely there was a student who had this. Okay, and let's see how we are doing on time. Okay, still there's time. So I'll just go through this real quick. So this was something I wanted to address. Okay, someone asked a question about earlier lecture on DNA replication. Moinak, I think. I don't know if I'm pronouncing the name correctly. So the question is that in non-Rs DNA containing vectors, very few transformants were formed, which is obvious as the plasmid lacks Rs, the autonomously replicating sequence. And we had this experiment where we were trying to identify the replication origin. So there was an experiment where you clone in different segments of DNA into a plasmid which has got a selectable marker. And the ones which have the origin, when you transform them in, they give more transformants because they are able to replicate by themselves. And so therefore those sequences were called autonomously replicating sequences. But in the ones where there were no RSs, definitely it's correct what he's saying, that there were a few colonies. And so the question is, how even a few cells survive without having Rs in their chromosomes. So this happens because if you take those transformants or colonies and you analyze them, that why is this coming? Why is it showing the selectable marker? Once it's growing, it means that marker is there and it is maintaining it. But it didn't have the replicative sequence. So when people analyzed that, they found that the DNA was there, but actually it had integrated into the chromosome. So then the marker is getting expressed in the chromosome, and because the chromosome is getting segregated, it's stably maintained. So this does happen at some frequency when you transform in DNA. And particularly in yeast, homologous recombination frequency is high, which is why it was a very good model system, especially when, in earlier days, it was very hard to knock out genes in other systems. And in yeast, we could do it relatively easily because the frequency of homologous recombination innate is high. So you put in a linear DNA fragment or even one which doesn't have um, such a sequence, so it will go and recombine and insert into the genomic DNA, and thereby it will be maintained. Okay. So next is, I think there are some questions for the guests also, the guest lecturers, and I've not looked at them. Maybe we can address them next time. But I think I wonder if one of the TAs has a reply to some of the questions on meiosis or one of the other questions. Yes, Swagat Nath or Debalina, would you like to comment or contribute on the questions related to meiosis? Or any other point you all want to add? I'd like you all to speak also. I've been going on and on.
Hello?
Meantime, I'll read some of these questions that students
Hello?
are writing.
Am I audible? Audible?
Yes, you are.
Yeah.
Great.
So we have some of the questions from meiosis that can be answered. So one of the question is during SE disassembly, if one of the daughter cells does not get any chromosomes, then does that mean that only one of the daughter cells can survive? Or is there any possibility that both the daughter cells can survive? So we have one of the live example where it is, for instance, in humans we have a Down syndrome where 21st chromosome we have a 3 copies instead of 2, like 2n plus 1. So that is one of the like direct example where segregation of chromosome and causes any like chromosomal defects, and you can actually see any number of physiological defects in the born child also. And that is one of the questions that I wanted to answer. And do all kinds of errors lead to aneuploidy? If no, what kind of errors lead to aneuploidy cells? And to answer these, like aneuploidy is basically because the same. It's basically the same answer where yeah. It's basically the same answer where if there is a defect in segregation of homologous chromosomes, then yeah, this aneuploidy basically occurs, and this
Yes,
basically
because there's another
this
session.
happens during the like diploiding stage. So that is one of the answer that, I mean, both of the answers are same. And there's one question: what is the function of Pcna? It's basically it's other name for beta clampin, uh, like eukaryotic cells. So that is one of the answer. It's a direct answer, to be precise. It basically holds the DNA with the polymerase, so that the DNA and the polymerase will not get separated, and so that the processivity of the polymerase will be increased with the help of Pcna.
Okay, I want to address one question, which meantime you can look up the next question, Swagat. Yeah.
Yes,
So Shubham is asking, I think it was asked in the forum also, and maybe it's an assignment question and students want to know about it. Shubham is there, very good. So the question is, what will be the effect of phosphatase inhibitors on nuclear envelope reassembly in telophase? He wants us to discuss about it. So basically this refers to, if you remember, there's an important phosphatase which works in the exit of mitosis, CDC14. So it refers to that. Basically, phosphatase activity is important, right? And we have discussed about laminin proteins, which were, when they are phosphorylated, that's when the nuclear envelope disassembly happens, right? So for reassembly, you want the phosphate to be removed, and for that, the phosphatase is important, which is anyway available and active in the exit of mitosis near telophase, where nuclear, after telophase or during that, the chromosomes have segregated and the nuclear envelope, it has to reform. So the phosphatase CDC14, which is a part of it, I have discussed the mitotic exit network in one of the lectures. You can look up that. Okay. So basically, if you add a phosphatase inhibitor, then that phosphatase will be inhibited. And so it won't dephosphorylate the lamins. And if you don't dephosphorylate the lamins filaments will not be formed. And that is important for the formation of the proper nucleus. And also, I think it was also mentioned that there are certain other nuclear pore proteins and various other proteins related to the nuclear envelope, which were phosphorylated, and then they are dephosphorylated by this phosphatase. Okay, so that's the answer, that if you use a phosphatase inhibitor, if it's actually targeting CDC14, then you will not get the nuclear envelope reassembly. Okay, so it's good that you have clarified your doubt. Is there any other main issue? There are some questions. I think they will be left. I think meiosis questions, we can address them next time also, unless Swagat has already some answer available. And they have posted some, the lectures which are of this week, probably, I think you all will be asking questions about them by the next session. So we'll visit those at that time. Some people are asking about the questions, it's a objective type exam, multiple choice type, the sort of how the assignments have been. So, okay. So the question, they'll be of a similar type. Now, what else is there? Someone mentioned molecular scissors. I think, that's a term popularly used for restriction endonucleases, or, enzymes that will cut the DNA. I mean, it's not very accepted, but it's a cool term to use. So let me just check my notes and see if anything I forgot to tell all of you. There are additional questions. So a lot of people were asking about Pcna and senescence earlier. So we'll look into this next time. And Swagat, you have any further questions to address related to meiosis? So we just have a few minutes, just 3-4 minutes are there.
Usually phosphorylates the retinoblastoma protein. Sorry. Yeah, PRB. So I can't share the image right now, but the answer is cyclin E CDK protein, which actually gets activated by P21. So that will phosphorylate the retinoblastoma protein, and once the retinoblastoma protein is activated, then is phosphorylated, then the E2F becomes activate. That leads to G1 to S phase. For reference, you can check for Leninger biosignaling, where the entire pathway is clearly explained.
Right. I'm also trying to open. There's a very nice graphic that I made, rather complicated figure that I have also made regarding that. And it's there in today's lecture also. So I'm just trying to open that and share the screen, because that's the best way, I think, to explain that. Yeah, here it is. So I shall share my screen, okay, momentarily. And I think very soon we may have to close the session. There's hardly 3 minutes left. So this is the one, yeah. So the students who asked that, they can take a look at it. So this is the graphic. regarding mitogen stimulation of cell cycle entry. So here you can see that the phosphorylation of RB is happening by the activity of this G1 and the G1S CDK. And what is the consequence of that? That the phosphorated RB, it cannot bind E2F. So normally it binds E2F and it represses the expression of these E2F dependent genes, which include cyclin E and cyclin A themselves. So when RB is phosphorylated by these CDK4/6 complex with cyclin D. Okay, so that's the complex which phosphorates RB, and then something's coming, I see, from the G1S CDK also. So it helps in this transition where the S phase genes will then be expressed, and it can help the cell move forward. You know, RB is a tumor suppressor protein. So as long as it's there, it restrains cell division. But when it's not there, then the cell cycle progression happens. So it not being there is equivalent to it being phosphorylated, because then E2F is free and it can go ahead and activate transcription of these S phase genes. Okay, so I hope that's clear. All right. My main request to all of you is to stay tuned. Okay. I actually did not see any questions from YouTube. I don't know if there are any pressing questions here, the YouTube channel. We can address them next time. Oh my God, there are a lot of comments. So I'll go through them and we can look into it next time also. Good afternoon, everybody on YouTube. I'll go through them. We'll save these chats and then we will some are the same questions. Okay. So my sincere request to all of you is, okay, time's up. So just please stay tuned and check the discussion forum. Thank you all for coming and good luck. Please join next time. I think we will end the session. Okay, TAs, everybody, administration.
Yes, ma'am.
We are ready to close the session, and we have the request to the students to keep tuned to the discussion forum and check the announcements. Okay, we will have one more session and we'll also be resolving your doubts. Thank you. Bye-bye.